Hi friends, you're back with me, your friend Professor Kukreja, right? So yes, we are preparing for NEET and we are preparing for biology. So here you can see some genes, some polymerase, some coding, transcription. Yes, we are with the molecular basis of inheritance, right? Question seems to be like the one which appeared in 2021 NEET, right? Okay. So identify the correct statement among the following. So let's focus on what is right in life. Also, let us focus on what is right and ignore what is wrong. Okay, let's go with it. So the split genes, right? The split genes, the genes which have the sequences which have some coding regions which we refer to as the exon and some non-coding regions which we refer to as the introns. And in the process of splicing, we remove the introns and join the exons, right? So okay, so that is split gene. So split genes are the characteristics of prokaryotes. Oh my god, no. The split genes is our characteristics. We have many unnecessary things. Anyways, so let us remove those unnecessary things and bring together necessary things. Okay, so the coding things, they are brought together and that is the characteristic of eukaryotes and not prokaryotes. So this goes out of the order. The capping, not the cap जो आप टोपी पहनाते एक दूसरे को, not that. Now it's sunny, so you can wear a cap. Anyways, so capping here we are talking about of hnRNA, the heterogeneous nuclear RNA, the one which is transcribed by RNA polymerase 2, if I'm not wrong, in eukaryotes, right? So capping, which will later on give rise to mRNA. Okay, so capping, it includes the methyl guanine triphosphate addition at the 3' end of hnRNA. Oh no, 3' end has a tail, right? We know that this is splicing and then there is tailing, and this tailing of poly A, it takes place at the 3' end, right? The capping, it takes place at the 5' end. कुछ भी बोलते हैं, right? So capping, it's wrong. So capping, it doesn't take place at 3' end, it occurs at the 5'. And now I told you that tailing, it occurs at the 3' with poly A tail. You tell me with how many residues? 200 to 300? Okay, you just tell me whether I'm right. Okay, RNA polymerase uses rho factor for the termination of transcription in prokaryotes. Yet uses, oh, this seems to be right. We know that to start the transcription in prokaryotes, it uses the sigma factor. So the sigma factor, it is used for initiation, and the rho factor, it is used for termination, right? So RNA polymerase uses rho factor for termination of transcription. It's right. But we'll still see the last one. The coding strand of the transcription unit is the one that is copied to mRNA. We know that unfortunately coding strand does not code anything. Though we label all the things related to coding strand, but we know that out of the 2 strands of DNA, the one which is the template strand, right, which is running from 3' to 5', that is copied to mRNA. Coding strand of DNA which is running from 5' to 3', it does not code for mRNA. So remember, coding strand does not code for mRNA. It is the template strand that codes for what you call as the mRNA. Right. So the right statement here is RNA polymerase uses rho factor for termination of transcription. Please tell me what it uses to initiate it. Okay. I just told in the earlier video. Not in the earlier video, in this video earlier. So number of chromosomes. I'm coming to the next question. I'm not wasting your time. Number of chromosomes in the anther filament is 16, right? So you have a flower which has a filament which has the anther lobe. So uska filament mein, in this filament, it has the number of chromosomes 16. We know that a filament is diploid. So you can say that 2n is equal to 16, right? So here is the number of chromosomes. So you have to find the number of chromosomes in the antipodal cell of the ovule of the same flower. I'm glad they said the same flower. So in the same flower, you have these antipodal cells, right? The embryo sac. Yes, we know that is the egg cell, the synergids. Then you have these polar nuclei forming the secondary nucleus. Then you have these antipodals. And you all know that these particular antipodals, they are haploid. They are haploid. So if 2n is equal to 16, oh, we left mathematics because we love biology, but then still we have to do some mathematics. So if 2n is equal to 16, n will be equal to 8, right? So number of chromosomes in the ovule of the same flower would be 8. Stay tuned with me, Professor Girish Kukreja, for more in biology and NEET. Thank you so much.